All right, let's move on to something I feel like you're a little bit more familiar with. Let's talk about enzymes. We are actually now on questions 15. What are the 4 main features of enzymes that you need to know? First of all, enzymes have 4 features. 1, enzymes speed up reactions. They are catalysts. They make reactions occur up to 10 billion times faster than they would without. You cannot live without enzymes. So enzymes speed up reactions. Number 2, enzymes can be reused. You use the same enzymes over and over and over again for all the reactions that your body has to carry out. Just like I keep saying this lock and key model, just like I use the same key every day to start my car, you can use the same enzymes to start many reactions in your body. So enzymes can be reused. They're not changed. They're not altered. You don't change the shape. If you change the shape of an enzyme, then it's not going to work anymore. 3, enzymes can recognize both reactants and products in order to catalyze in both directions. Enzymes can recognize both reactants and products in order to catalyze in both directions. And then 4, enzymes are very selective about the substrates to which they will bind. I talked about this the other day. I cannot take the key to my car and start my husband's truck. Enzymes are very specific to what they bind with. They only work on certain substances. If you remember, a substrate is what our enzyme binds with. I remember the activity that we did in Biology 1. We went out into the commons area and there were puzzle pieces all over the wall, and you had to match. You had the enzyme in your hand and you had to go around and find the substrate on the wall to see where they fit. The certain enzyme fit the certain substrate, and when you join those together, they react. Okay? So now let's move on to number 16. How do enzymes increase the rate of a reaction? We talked about this a little bit a minute ago. They increase the rate of the reaction by lowering the activation energy. They're going to lower the amount of energy that it takes to get a reaction started. Okay, 17. What is an active site? What is a substrate? Okay. Active sites are the areas where an enzyme binds with the substrate. Okay, and the substrate is just the area, it's what an enzyme binds with. Hold on, and I'll actually draw you a picture of this. So it says enzymes have active sites, which are areas that allow a chemical binding of the substrate to the enzyme. So it's essentially the keyhole. It's where they fit together. So now I'm going to do this, and I'm going to show you what this would be. So let's just say that this is the enzyme. Okay? Well, this right here would be the substrate. Okay, it is what an enzyme binds with. This area right here where they join is called the active site. Okay, like a lock and a key or like a puzzle piece. That is also what is meant by that induced fit model. Okay, here's a better diagram than my drawing. You can see here in step 1 it says the substrate binds to the enzyme. So here's the enzymes called sucrase. Alright, and here's the substrate. It's going to bind together, and when it does, we start a chemical reaction. So look at number 2. Substrate is converted to products. 3, products are released. We actually created glucose. And then 4, the active site is available for another molecule of the substrate. So the active site is available right away again for another reaction. Okay, and that leads us to number 18, which I kind of already have been over. A lock and key model just is, you know, the model that says the enzymes fit with their substrate like a lock and a key. They have to have a specific shape. Okay, and look at 19. It says, what suffix do most enzymes end in? Well, just like this sucrase, enzymes normally end in the suffix ase. Some examples of other enzymes in your body are carbonic anhydrase. That is an enzyme that your body has that speeds up the rate at which you get rid of your carbon dioxide. You have an enzyme in your body called lipase that helps you break down lipids. You have an enzyme called lactase that helps you break down the sugar in milk. Okay, so there are a lot of enzymes that you have, and most of them start or will end with the three letters ASE. Okay, so let's look at number 20. What are the three main factors that affect enzyme activity? Okay, so as I'm reading this, I'm thinking about the lab that we did the other day with the toothpicks. So if you'll look at this, first of all, I would say that temperature most definitely decreases enzyme activity. I've got a typo there. You'll have to ignore that. Temperature decreases enzyme activity. Not only high temperature, like it says, but very low temperature, too, decreases enzyme activity. I think about, Brandon's and Morgan's and Tyler's hands. Well, except for Tyler, he was a little different. It definitely could not break those toothpicks as fast when your hands were that cold. Remember, that's why we put we put food in refrigerators, because at cool temperatures, the bacteria's enzymes cannot work properly, so they can't spoil your food. But the same thing also works for really high temperatures. If it's a really high temperature, then the enzymes do what we call they denature, and they don't fit anymore, and so they can't work. Okay, so there's temperature. Also, pH affects enzyme activity. Your enzymes work best when you have a pH near 7, which is neutral. Your body doesn't need to be too acidic or too basic. And also, salinity. It says all enzymes have a certain salt content that they like to work in. So those are all factors that affect enzyme activity. Temperature, pH, and salinity. Okay? If you look at number 21, do enzymes prefer high or low temps? And basically, neither. They just kind of like moderate temperatures. Okay? And then number 22 says, what is the ideal pH for an enzyme to work? Well, it's best near 7. All right? Let's look at a couple of other terms. There's a term here called cofactors. Remember, co means both, and usually co means that you're going to work together to do something. So cofactors are things that actually help enzymes. So if you look here, it says cofactors are metal ions or coenzymes that bind to many enzymes and make them more reactive. Okay, so they actually help enzymes work better than they would without them. Okay, so that's number 23. If you look at number 24, it says describe 2 examples of cofactors. I'm sorry I didn't leave you enough room there. But for 24, it says cofactors are metal ions or coenzymes that bind to many enzymes. I'm sorry, I messed up. Examples of cofactors are antioxidants like vitamin E and catalase. They work to help neutralize free radicals. And then you also have inorganic metal ions such as iron that serve as cofactors in the processes of the chloroplast and the mitochondria. So you have cofactors that actually help your body carry out processes in the chloroplast, not really you, but plants, carry out photosynthesis and cellular respiration in mitochondria. So you do need a lot of the minerals that you can get from your food and things like that. All right, and then let's look at number 25. Describe the process of bioluminescence in terms of enzyme activity. We're going to skip this one. Okay, so we all have heard of what bioluminescence means. We know that's when organisms can give off their own light. It says fireflies and many other organisms use enzymes, what we call luciferases, to produce light by bioluminescence. Okay, and that actually is caused by the action of an enzyme. So let's look here. It says reactions begin when ATP is in the presence of oxygen, which transfers a phosphate group to luciferin. And when that happens, electrons in luciferin become, that should be excited, not exited. Let me change that real quick. Okay, so electrons in luciferin become excited and begin reacting, which releases fluorescent light. anytime you give off any kind of light, that's when electrons are being given off. So that's just the ability of organisms to give off their own light. Okay? There are many creatures who are bioluminescent. Think about the ones that live in the very deep oceans. And there are also many, on land too. Okay, so that actually covers our enzymes and our energy.